Satellos is a biotech firm developing therapeutics targeting degenerative muscle diseases. We've got CEO Frank Gleeson and CFO Liz Williams joining us. They're here to discuss the recently started phase 1 trials and the company's path forward for 2025 and beyond. I'm Martin Gagel with Market Radius Research. It's Thursday, October the 3rd. Please remember this is neither a recommendation nor investment advice. We're here to learn about the company. Frank, Liz, welcome back and thanks for joining us. We last talked in May when you gave a broad overview on Satellos, your technology, and the path forward. You've accomplished a lot in the last 4 months, including some remarkable results from animal model studies that were just released, and you've begun dosing your first patients in your phase 1 clinical study. Welcome, and why don't you bring us up to speed on what you've been up to?
Well, super. Thank you very much for the introduction, Martin. And it's always a pleasure to meet with you and to meet with your audience and the many shareholders of Satellos who may be listening in. So welcome to everyone on the call as well. In this call, we'd very much like to provide a bit of a refresher and an update on Satellos. So we have a few slides to walk through to that objective. And as Martin indicated, this is interactive, so we welcome questions and hope that there are many questions. So as we move along, we'll first off remind you that as a publicly traded company, we may have forward-looking statements and to take them under advisement. So as a quick refresher, our company is built around a very novel idea, the idea of a pill that can regenerate muscle in tablet form, as exemplified on the slide. Satellos 3247 is our lead molecule, and it is delivered as a tablet. It is based on disruptive, game-changing science from my colleague, Dr. Michael Rudnicki's laboratory in Ottawa. We are addressing unmet needs in markets that have multi-billion-dollar potential. And we have a track record of delivering on what we promised to do. And we have many upcoming catalysts over the next 12, 18, and 24 months. So just, again, a quick refresher on what we see as the corporate highlights of Satellos and what distinguishes us. We have a novel drug with a very clearly differentiated mechanism of action and approach to treating disease. We're now a clinical stage company, which for us is a huge milestone and a big step forward. Our lead program is in Duchenne Muscular Dystrophy, a fatal disease for which there is no cure and no disease-modifying treatments. We operate with a significant valuation gap to our peers, which on the one hand is a bit infuriating to me as CEO and to many of us as management, and I know some of our shareholders. On the other hand, the upside is just extraordinary. We've put together a very accomplished team and very, very, very good investors of the highest pedigree on Wall Street and across the US. Next slide. So the opportunities in muscle regeneration truly are significant. So in the muscular dystrophy space, as I mentioned, our lead opportunity is Duchenne Muscular Dystrophy. The US prevalence is 20,000 patients. That means 20,000 people have this condition at any one time in the United States. The statistics are the same worldwide. So take the ratio of 20,000 to the population of the US and multiply by any country you have an interest in knowing about. And as I mentioned, it's fatal. The market for drugs for dystrophies is projected to exceed $20 billion. That was recently outlined in a report on muscle by the research group at Cantor Fitzgerald in the US. We can provide that if you wish to see that. We can send you the link. And the next market area in muscle trauma is also a very significant area. 66 million people visit trauma centers in the United States annually. costing the US system almost $200 billion. And again, it's an area where the treatments are very costly, they're ineffective, and the need is quite great. And finally, another area that we believe our approach will have utility is in aging, both in cancer and non-cancer aspects of aging. Cachexia is the muscle loss associated with many cancers and causes deaths in many cancers, as opposed to the cancer itself. And then, of course, as we age, many of us experience muscle weakness and muscle loss. And again, this is an area that's wide open for new treatments. So since we last spoke, we have been busy, and we've done what we said we would do. We completed our IND-enabling studies so that we could apply for and initiate clinical development. We manufactured our drug under GMP conditions in sufficient quantities to satisfy our needs for the next 2 to 3 years, depending on how many trials we decide to do. So we have several kilograms of product. We have achieved regulatory approval and have initiated our phase 1 study in healthy volunteers. We have achieved rare pediatric disease designation in addition to orphan drug designation. The rare pediatric is the one that comes with a voucher that potentially has a significant value, currently $100 million or more. We've dosed our first patient, and not only that, I can update you today to say we've completed our first cohort. So we're moving from our first dose level to our second dose level, and you can expect that we will do this now on a regular basis moving forward on roughly a weekly cycle, 1 to 2 week cycle. And we have this week announced what we consider to be, and what feedback is suggesting to us is can accurately be described as groundbreaking data. And we'll go through some of that in a little more detail on this call. And just as a reminder of the valuation gap, which putting this slide up hurts me, at the same time, it really does indicate the valuation potential and the valuation that the capital markets are willing to place on treatments for Duchenne and other dystrophies. This is an area that is seen in the financial community and in the medical community as one of enormous need, and it has the interest of Wall Street. And that's a good place to be. And of course, our job as management over the next period of time is to close that gap, which we hope to do by continuing to make progress. Next slide. And so what makes us different? We've tried to capture this because we find in talking to investors, whether they're retail investors or whether they're Wall Street investors, whether they're casual investors, whether they're highly sophisticated biotechnology investors, that this is a confusing field. And sometimes people don't quite understand the differences. And this is something we also will be working on more and more to make sure people do understand the differences. But let's start with our drug, 3247. Number one, this is an oral daily pill. It can't get more easy than that. It restores regeneration, and by doing so, that can become disease modifying. In other words, we bring muscle back. We make new muscle. Our drug is designed to have a wide safety called a therapeutic window. Our drug will not be restricted by the mutation status or whether the child is ambulatory or non-ambulatory. In other words, all patients. And it's intended to be able to function as standalone therapy or to be adjunctive therapy. So a child who has previously received a gene therapy treatment will not be blocked from using our drug. In fact, it may have synergy. Now, what is gene therapy then? How does that work? Well, gene therapy is designed to replace a portion of the dystrophin that is missing in muscle fibers to stabilize those fibers. In other words, to, at best, keep the disease from deteriorating further or from slowing the deterioration by trying to arrest the progress at a certain point in time. It's a one-time only hospital-based infusion. It often requires an immunological preconditioning. So it requires a stay in hospital. It does not make new muscle. Biologically, it is not designed to make new muscle. And biologically, it does not make new muscle. And it comes with the potential for safety and immune concerns in a number of the patients. And the product that has been approved is priced at over $3 million. So that is gene therapy. I'm not saying it's bad. I'm not saying anything other than the facts as they are. Now, what is exon skipping? This is also a genetic approach. And in this case, the idea is to build a bridge over a mutation that occurs in the gene itself, where a particular exon may be missing a component. So exons are the coding regions of a gene that allow a protein to be made. And there are a handful of mutations that appear to occur regularly in boys with muscular dystrophy. Altogether, this handful account for less than half of the population of Duchenne boys. This is a treatment that also is an infusion and is given on either a weekly, now moving to less frequently, say monthly infusions. It also has the potential for safety and dosing concerns because you are injecting foreign substances into the body. There is no evidence that we have seen that in any way suggests that this approach creates new muscle. And of course, it's limited to those patients with a specific exon mutation. So every different mutation requires a different product. And as I mentioned, that is less than half of the population have the handful of mutations for which exon skipping could be developed. So we think that we have a very attractive offering to the medical world and to patients that could be adjunctive to gene therapy or exon skipping, or standalone, whether a child receives or does not receive either of those approaches. So this is why we're so bullish and excited about what we're trying to do. So this week, we issued a press release on data that we are presenting at the largest muscle conference in the world, which occurs once annually this year in Prague, called the World Muscle Congress. And so this will literally be the who's who of all the top research people in the world and all of the top development programs in the world targeting muscle. And of course, there's great interest in Duchenne muscular dystrophy at this conference. So over the last couple of years, we've repeatedly been asked, have you done a study in the canine model of Duchenne muscular dystrophy? And the answer now is yes, we have. And this is also an important step because it's widely considered in the scientific community, and frankly in the drug development community related to Duchenne, that this model, the canine model, is a higher bar than the mouse model, than any of the mouse models, and it more accurately reflects what happens in a human being with muscular dystrophy. And largely that's because the mutations that occur in humans spontaneously to create this disease occur spontaneously in dog. So this is not an engineered muscular dystrophy the way it is in a mouse. This is a naturally occurring spontaneous mutation. And then colonies of dogs with muscular dystrophy can be bred and studied. Now that makes it very, very difficult to gain access to these animals. And there can be very long lead times to get into a program to work with these animals. So we were fortunate to have a relationship with a professor at a major institution in the United States who was able to make 2 animals available to us to do a 4-month study of our drug in those animals. These animals were relatively old for this disease state at the outset. They were close to 9 months of age. They were basically 8 months and 3 weeks, and we treated them for 4 months. Typically, others that have done studies in these dogs typically have started with much younger animals. But as I said, we were able to access these animals, and so that's what we worked with. And we looked at many different parameters of these animals. What I'm going to show you today are 2 of the most important aspects: the effect on the muscle itself and the effect on the strength of the muscle. And we believe that if you can affect those 2 items and not cause any side effects of significance, that's a big win. So the next slide, I'm going to show you images of untreated and treated hind leg biceps in the dog model, in one of the 2 dogs. And I will preface it by saying that we have many, many, many of these images in both animals and in different limbs, and it's a consistent pattern. So typically, a Duchenne animal at about 9 months of age, as depicted in the far left picture, has muscle looking like that picture. The red that is stained for, which means it's a fluorescent dye that basically tells us what we're looking at, is stained for a flag, an immune flag, an immunoglobulin factor called IgG, that signals that there is dying tissue in the cell. And you can see the animal that we worked with, which is one to the right. We see that same pattern. In fact, it probably looks worse than the one on the left. But the reason we want to show you these pictures is so you understand we didn't start with an animal that was in really good shape. We started with an animal that was in really bad shape. A lot of dying tissue as depicted by that red. So you're looking at a slice of the bicep, and all of that red is all dying muscle tissue. Now we treat it for 4 months. And what you see next, okay, is a complete change, what appears to be a complete change in that muscle bicep. All that red representing necrotic dying tissue has been corrected. And on the right side, what you see is what a normal, healthy 13-month-old bicep in a dog looks like. And those pictures are very similar. Now, we have not made the muscle perfectly restored to 100% health. We have restored this muscle to the point that there is more new tissue being created than existing tissue dying. We have changed the course of the disease in this dog. That's how we interpret this information. And of course, this is meaningless if it doesn't lead to a stronger muscle. At least that's our view. This is a pretty picture. So what happens to the strength of these animals? And here we've graphed it. So looking at that bicep and looking at the torque in that muscle that it can create, which is the sort of gold standard measure of strength, you can see both animals starting at 8 months and 3 weeks of age, where we first started treating, over the next 2 months where we took measurements, and then over 4 months where we took measurements, that the strength of those muscles has increased quite dramatically. And when we measure all the limbs together, at 2 months, we increase the strength 111%, and at 4 months, 195%. These results are as good as a normal dog and better than has been reported with gene therapy. And we're always asked, how do you compare? So on the next slide, we'll show you how we compare. And again, some graphic information. So I'll just take a moment to describe the graph on the left. What we're looking at here is our data, our same data in the teal and the mauve, superimposed on a graph, which is showing what typical DMD dogs and what typical healthy dogs look like in terms of strength. So the strength scale on the y-axis is the same for all 4 of the measured entities: healthy, diseased, and our 2 treatment animals. And this is based on published data in the literature by Dr. Cornegi, who is the leader by consensus in the field of gene therapy and Duchenne muscular dystrophy dog models. So the black dotted line depicts the typical pattern in, Duchenne animals. So from birth, or in this case, it looks like the axis is set at 3 months, through 6 months, you can see the animal is increasing in strength despite having Duchenne muscular dystrophy, and that's because the animal is growing. It also means that the animal, despite having no dystrophin, is able to actually have functional muscle. As the animal gets older, it stops getting stronger. And then after 12 months of age and beyond, it begins to go into decline. A healthy animal, by contrast, keeps getting stronger. And you can see that we've taken our 2 animals, which based on this slide, this graph, were below average in terms of strength to start with. So we weren't working with healthier than normal Duchenne animals. And we've restored them basically to normal in terms of the strength of their muscles within 4 months. This is truly a remarkable achievement. And then when we look at what's been published by the experts, again, the publication is cited. We superimpose our data on data published for gene therapy in the same dog model that we used. In this case, the x-axis is not the age of the animal, it's the length of the treatment time, okay? But the y-axis is the same strength measurement, okay? So it's the newtons of strength that are generated based on the amount of muscle and the weight of the animal. So what you see here, again, the same pattern with the Duchenne animals. They don't get stronger. The untreated Duchenne animals do not get stronger. That's in black. The treated animals, treated with gene therapy, do show that they're stronger. after 90 days of treatment. Our animals show we continue to get stronger and get more strong than the gene therapy treated animals in this case. But what's really significant here is that the animals that were treated with gene therapy started being treated when they were 3 months of age. And then they were measured when they were 90 days later. And so what you're seeing is that over that 3-month period, these animals are naturally getting stronger. You can see that on the left, that animals that are Duchenne animals naturally get stronger between 3 and 6 months of age. We started with dogs that were basically 9 months old, that were already severely injured and in decline. So we think that this is pretty compelling evidence that we have a potential treatment for this disease with the prospect of making a disease-modifying difference. And again, I'm not putting any of this information forward to criticize any other approach. I'm simply talking about the biology of regeneration and what it means if we can create new muscle. If we can create new muscle, it will be stronger and it will make a difference. And that's what our drug has been being designed to do. So,
Frank,
yeah.
I'm curious, did you get any evidence from the researchers that, let's say, the dog's behavior, that they could climb upstairs better, or that there was sort of a physical manifestation of their healthier muscle?
Yeah, so we monitored the drugs, or the researchers monitored the animals over the time period in which we were treating them. They were kept in a kennel, so they weren't, this wasn't, American Kennel Club where they were out jumping through hoops and all that kind of stuff. And so, their gait was measured, the amount of activity that they used was measured, and generally, and all of that is in the poster that will be shown in Prague and is on our website, I believe. And what you can see is that very clearly these animals did not get worse in terms of all of those characteristics. In fact, overall, they look to be a little bit better overall. So it would say to us that they were a little more agile. But remember, these animals typically, over this time period, decline very, very rapidly. So strengthening their muscles so that they can function and continue to function is a very big win. So this is our upcoming plan. There are basically two parts to it, I suppose. In the upper panel, you're looking at our program that is underway now in Australia. And we, just to remind you, did our work in Australia for 2 reasons. One, because there are massive tax credits of over 40%, which is direct cash rebate for doing work in Australia. But 2, they have established a regulatory system that feeds very nicely into the US system, into the FDA, with standards that are equivalent, and provides flexibility to allow sponsors to design trials and get sort of pre-approval for various steps, which helps to telescope time. And in this case, our study will have 2 broad components. The first component is the safety and pharmacokinetic study in healthy volunteers, which is underway now. And the second is to move into adult Duchenne patients, also to do safety and PK work and some exploratory biomarker work. And then in parallel with all of this, we are conducting additional toxicology work that's required before we can go into pediatric patients. Those would be children. In our case, we will be treating children that are ages 7 and above. And so we need that tox work to be done to allow us to go below adults age-wise, and then we will plan to begin our clinical program in those children approximately the middle of next year. And so that's laid out here. So that basically means we're going to have a lot of news flow over the next year as we move through our various studies and we initiate new studies and we have data coming out during those studies.
All right. And just, sir, if you can
Mhm.
go back there and just to quickly clarify. So what you've started is initially is the trials for safety and tolerability in healthy
Yeah,
adults, and that continues on through, let's say, the end of January or so. And then in December, you start recruiting, and early next year, administrating on Duchenne's patients themselves to see
correct.
how
Correct.
it affects them.
Absolutely. That's it. You've got it exactly. And then thereafter we will go into children, and we will do a proof of concept study in children in which we will be looking to see if we can affect their muscle and the strength of their muscle, analogous to what we saw, quite frankly, in the canine model.
And with the children, roughly when would we expect to have that beginning?
So
That's the bar on the bottom line, the sort of
correct.
beige, and then the
Yeah.
purple blue one.
So the beige is the toxicology work that
Okay.
will wrap up um, at about the end of June, when we have all of the final reports from the contract research organizations that we could submit to the FDA. Then we submit to the FDA, and then we begin our trial during the summer of next year.
Okay. And that is like a phase 2A. So you're looking for obviously safety and tolerability, but you're looking at
Right.
effectiveness in
Yeah.
part
So
as well.
yes. So those are good questions. This will be, of course, all trials in terms of the FDA have to be safe. So we always have to design in a safety component of trials, particularly when we move into children. But this we're designing to be a proof of concept efficacy study. And so the more we can drive efficacy, the more potential we have to create an environment for accelerated approval, or consideration of accelerated approval. And we believe, based on what we've seen in the dog, that if there is translation from the dog to the human, we should be able to see effect. And that would separate us from everybody else, because that is the one piece that has been missing, is clear indication of strengthening of muscle. That's the one piece that has not been apparent. There have been some indicators of improvement and so on, but there hasn't been that kind of true demonstration that the muscle quality and the muscle strength have improved. And that's what we're going to try to show, along with using MRI to image the muscle and show how the characteristics of the muscle may be improving, to use biomarker readouts to see how we're affecting the immune system, because these children have, all of them with this disease, have immune system repercussions, because the body thinks it's under attack because of the dying muscle cells that occur that I just showed you pictures of. And we believe that we can ameliorate that condition quite dramatically. So these are the types of things that we'll be looking for. And as I said, we're not just hopeful, we're excited, and frankly, we're optimistic that we'll be able to see something, and that we should be able to see something within the time frame of a proof of concept study, within a 3-month to 6-month time frame. So we'll design this as a 3-month study with what is called a longer term follow-up, which will be an additional 3 to 6 months. And what we hope to see is efficacy. So we believe that that is what we have consistently seen, study after study after study. And in going from mouse to dog, we actually saw it on a larger scale in the dog than in the mouse. So we actually think that our mechanism we'll do better in larger muscles than smaller muscles.
With the current phase 1 that started and then the phase 1A on the Duchenne's patients, you've announced the enrollment of the start with the first person. Will there be any update data or information along that time, or will it be now you've dosed your last person or is there any sort of milestone or anything you can
Yeah,
comment on
we
during these
well,
phases?
I can comment today, and I can also comment on what we will intend to disclose as we move along. So thank you for the question. So first, we've already cleared our first dose. So we've completed our first cohort at our first dose level in our single ascending dose safety component. And the review panel, all of these studies have a medical panel that is set up, have authorized us to move ahead to the next dose level. So we'll be dosing those patients between today and the middle of next week. And so we will probably finish the second panel, the second cohort, within the next 2 weeks. And that will kind of be a regular routine, but we don't plan to press release every time we go from one cohort to the next. What we will inform the market of is when we move from the single ascending dose to the multiple ascending dose. So the difference being that a single ascending dose is one dose, so one pill in this case, for one dose level, just like you take one Tylenol, then we go up and we give them more pills so that the dose level goes up, but only one day. And that's because in doing these trials, you have to establish that you don't see anything even with such a low level of frequency. Then the next piece, when you go to a multiple ascending dose, is you repeat that kind of pattern. You keep going up with dose increases, but you treat each patient for a longer period of time. And in our case, that will be 7 days, because that's the predicted pattern for the pharmacokinetic properties of our drug. So that's what we're really looking for. The combination of the two of them gives us the safety and the pharmacokinetic coverage information that we need in order to establish dosing levels when we go into patients. And then when we move in, so we will announce when we move from the single dose to the multiple dose. And we may, of course, then at the end, when we have our data from all of the studies, we'll announce that. So yes, there will be interim updates, and there will be data coming out. If we're able to, these are blinded studies, so we have to be careful about trying to release data that is meant to be blinded, because we don't want to compromise the integrity of the study.
Right. If there would be, let's say, some unforeseen side effects or safety issues and things had to stop, that would then be announced, right? So essentially no news is good news while you're going through this process?
That's a very good way of looking at it, because if there was an issue and it was material and significant, then we may be stopped from moving from one dose level to a next, and we would have to let the market know that.
Yeah, we went through a lot there. Thanks. Yeah,
Yeah,
continue on.
so the first 3 check marks are a repetition of an earlier slide. And in addition, over the next few months, as we've just discussed, the fourth point is updates on our PK and safety studies, moving into Duchenne patients in adults for a PK and safety study, completing that study, and then completing our toxicology and filing to begin a proof of concept study about mid-year, as was on the previous timeline slide. And then I think at this point, I think we must be close to the end of slides. I think that's a good one. Yeah. So I guess this is the summary of, just to let you know how we're talking to the market and how we're talking to other investors. We have near-term clinical milestones in phase 1 and phase 1B over the next 6 months. We are clearly in a space where there are very high valuations. We have a very strong team and great investors. And, the markets are really quite significant. And, we'll be moving into phase 2 clinical development in a short period of time. So that's kind of an update, and happy for questions from the floor.
Excellent. All right. We've gotten several questions from the audience here, so that is much appreciated. I'll start at the top here. I would like to know the timeline of potential approval if all the data is positive, and I guess he's talking phase 1, 2, 3, and final approval there. What would that if things work out? Obviously, lots of variables, but in sort of a normalized timeline, what could people expect?
Yeah. So everything depends on, data, magnitude of data, and of course, the receptivity of the regulators to a dialogue. So typically, it does take many years to get a drug approved, even an orphan drug, and it wouldn't be unexpected for a Duchenne drug to be a 5-year process, certainly a 3 to 5-year process. What we're trying to do is develop proof of concept data in a well-powered study, which is the phase 2 study, that is designed to show an effect both on the physiology as well as the function of muscle. And if we can do that, we believe, given the urgency of this disease, given the urgency of treatment, because muscle is lost every day for these children, literally they lose their muscle. It doesn't just weaken, they actually lose it. That we would have a very good argument to discuss with the FDA and with other regulators the potential for accelerated approval. And so we hope, and we are planning, and we are designing our trials to put ourselves in that position by the end of 2026. So that's 2 years from now. So that is what we're trying to do. But that is a plan. That is our objective.
You've got rare pediatric disease designation, and included in that is a voucher that that has potentially some significant value to it. Could you just dig into what the rare pediatric disease, what that means in this voucher, and how that whole process works? 'Cause I think that sort of dovetails in a bit with potential accelerated timelines.
Yeah, so it really goes back to an objective of previous U.S. administrations and the FDA to encourage drug development in orphan or underserved areas, with a particular emphasis on childhood diseases. And so somewhere along the way, the idea of providing an incentive by essentially giving drug developers a lottery ticket, if you will, that they could use themselves or transfer to another party to get an early approval on a drug, or to put themselves in the queue or in the lineup for an early approval on a drug, would have value. So when we get our drug approved, that voucher will then have value. We can then sell that voucher to a third party or use it ourselves on another drug candidate to get a review of a drug for approval. So these vouchers over the last several years have been sold on the market, kind of, so to speak, and they typically are valued at 100 million US, and now that number seems to be trending up because the FDA is, or the US system is thinking about stopping this program, grandfathering those that are there and then stopping the program. So the value would go up.
And I guess that highlights the value of time in doing research, where you can, if you can accelerate things, that is of huge value to
Yes. So everything's about time. I mean, if a company is developing a drug that has the potential to be $1 billion of revenue, so that's $250 million a quarter. So 3 months is worth $250 million, right?
Yeah, yeah.
That's how people look at it.
Will there be a liquid form of the treatment for those that can't swallow pills, like smaller children?
Yeah. So the pill that so the answer is yes. In fact, the dogs received a liquid solution wrapped in bacon. So the challenge with Duchenne, of course, is that there are undoubtedly individuals that have difficulty swallowing because they're the muscles that are required for swallowing deteriorate. We've actually made a very tiny pill, so we don't anticipate that there will be any issues with young children swallowing the pill that we have designed. But if that does prove to be the case, then of course we will make a solution.
There's some questions here on the clinical trials, where they're happening, how they're happening. Someone's asking if they would be able to participate in a trial if there are clinical sites opening in Europe, particularly the Nordic countries. Can you, like, because this isn't just, well, all diseases are horrible, but this is very impactful on
Sure,
young children and families,
sure.
and raises some hope. And people want to, with good evidence like you're initially getting here, raises some hope. Can you talk about, yeah,
Yes,
trials, where
yes, I
they'd
can.
happen, how they would happen?
Yes, I can speak to that. So we're in the process now of designing our phase 2 program. So we have not locked down our phase 2 program. It's in design. We intend to have a multi-site trial. We certainly intend to be in a number of countries. Naturally, our company is based in Canada, so Canada will be one of those countries. The US will be one of those countries. We're already working in Australia, so we will continue to have a presence there. And then we will be looking at other countries, particularly we'll be giving consideration to Europe. Whether our very first trial is more of a North American focus, that will be determined. We have not made that decision yet, but we will be doing trials globally in time. So if it's not immediate, it will be shortly thereafter.
I don't quite understand this question, so I'll throw it out there and maybe it makes sense to you. Can you explain more of the method of action, sorry, and what cellular processes may be involved? Could your drug play a role in patients with eating disorders or other things where muscle growth is difficult?
The mechanism of action of our drug is to affect the process by which muscle cells are formed. So muscle cells are created in response to damage. So when muscle is damaged, the muscle itself asks for new muscle to be made. And it does that through a series of biochemical steps that send essentially a biochemical signal into muscle stem cells. Muscle stem cells reside deep inside the muscle fiber. And when they receive the signal that says, you're needed, wake up, so to speak, that stem cell then divides to create a new muscle cell, or the progenitor of what will become a new muscle cell, and to create and leave behind a stem cell so that there's a cell there in the future when the next call for action comes. What we discovered in Duchenne muscular dystrophy is that that process of sending the signal to the stem cell and then the stem cell responding, the response is impaired in muscular dystrophy. So after a muscle stem cell in muscular dystrophy receives the signal to divide to create a new muscle cell, it is not able to do so with enough efficiency to solve the problem. So gradually, what happens is the bodies of these children are unable to make enough new muscle cells to keep up with the ongoing damage that happens to them as a result of the genetic problem with dystrophin. So our drug has been designed to basically correct that issue, to essentially reestablish the ability of the stem cell to divide properly. So it is a method or a mechanism that we believe would be effective in any situation where muscle is damaged and not repairing itself properly.
On one of your earliest slides here, you talked about you had sort of three different target areas, and one was trauma to the muscle. And this is what you're referring to. So
Yes.
even if some in a car accident or whatever,
Exactly.
this could potentially early stage, not to get ahead of ourselves, but that is a potential idea of where this how this could treat people as well.
Absolutely. So where there's a situation that muscle is severely damaged, then the body will call on the pool of stem cells to repair the damage. And our drug is designed to enhance that process.
Do you have any ongoing dialogue with EMA? And I don't know what EMA is.
I'm guessing that that's a reference to the European regulatory body. At this moment, specifically, we are not in dialogue with them. We will be starting outreach in the coming months, but at this precise moment, no. But we do know them well. Our head of our regulatory group is very experienced with applications in the European community and has successfully had drugs approved for clinical development in the European community. So it's an area where we know well, and we will be there in the coming months for dialogue.
Question here, and I'm going to add on to it a bit. When do you see the market starting to value your progress? And is in the of drug development through the different phases of clinical trials, is there a point that typically where the market says, oh, okay, this was significantly de-risked and the potential is there? Is it after phase 1 or some particular type of data where the market really wakes up to things?
Well, of all the questions I'm asked, that's the one I wish I had the answer to. I'd rather go back to more science questions. No, just kidding. Yes, the market does value companies more, I suppose you could say, when they see proof of concept, unless, of course, the proof of concept they see is less than what they dreamed of. So in the case of Satellos, we try to understand this disconnect. We think there's a disconnect between the value we've created in terms of the progress of the company and in terms of the data that we're generating and the market reality of what our stock price is. I think part of it is, frankly, the Toronto exchanges aren't valuing biotech the way we could get valued on other exchanges, which is one of the reasons that's part of our planning. But more fundamentally, we are challenging the scientific consensus around this disease area. And so as we move along, sadly to us, but the reality is the burden of proof is on us. So, the dog, I think the canine data is already showing to us that the receptivity of large institutional money, large institutional investors is increasing for us. Now they're not open market buyers on the Toronto Exchange for companies with our market cap. So that's also a reality. But it really encourages us. The Wall Street banks are very excited. They're inviting us to all their conferences. They want to meet with us all the time and talk about plans and so on. So I think it's coming. I can't give you a specific date, but it's coming. And once we're in, once we're in a phase 2 trial and once we're generating data in patients, and as soon as we can show a signal in patients, then I think, we know what the values of all these other companies are. Um, it's, it's going to happen. It's going to happen to Satellos.
You've raised a remarkable large amount of money at a way above market value to fund this current phase.
Yep.
Could you, but developing drugs, as we all know, is very expensive. It doesn't take you down the whole path. Can you talk about your market strategy? You mentioned NASDAQ. What is your capital market strategy? How do you expect to fund the ongoing research and discuss your runway and so forth?
Well, I'm going to ask my key partner in this strategy and our CFO, Liz Williams, to address that topic, which is occupying a lot of our time.
Thanks, Frank. Thanks, Martin. Great question. As Frank mentioned, it's something we're thinking about all the time and working on all the time, meeting with lots of institutional investors. So as you know, we already have in Satellos 5 very large deep pocket investors that are quite excited about the progress that we've made to date. So we do anticipate that those investors will provide additional funding when we're ready for it. So our plan is to list on NASDAQ in the first half of next year. So we intend to be listed on NASDAQ before we start the phase 2 proof of concept study in Duchenne, pediatric Duchenne patients. The exact timing will depend on market dynamics, honestly. So if the markets look good, we may go ahead and do that sooner, but we think it's important to be listed on NASDAQ when we start that trial to really get the eyes on the stock or the validation for getting to that stage, which would be difficult to do only listed on the TSX. So we are fully funded through the current phase 1 study. So the healthy volunteer study, the adult Duchenne study, all of the toxicology work that's ongoing to support the phase 2 proof of concept. But we will need to raise additional capital to complete that phase 2 study. We anticipate that will, like our last financing, it will be US institutionally led. So it won't be a marketed offering where it's heavily discounted with warrants, et cetera. It will be a very tight financing led by healthcare specialist funds in the US that will hold for the duration of the study. And then we would anticipate realistically that might be the last financing that we do, because we'll have proof of concept data after that point in time, in which case Satellos would be poised for either a very significant partnership transaction that would provide non-dilutive capital or an acquisition.
All right. And would that financing be coincident with the uplisting to NASDAQ, or could you do the NASDAQ listing without a financing? Usually they go together.
Yeah, it can be either way. So we could do the financing first and then follow on with the NASDAQ listing. We can do them at the same time or within a couple of days of each other, or we can do the listing with a follow-on financing. So our preference is to do the financing follow-on NASDAQ listing or coupled together. Because we think it's important to have the institutional shareholders in the story when we list on NASDAQ so that they can provide liquidity and support after the listing. It also gives us an opportunity to include some US banks so that we have support from a capital markets perspective and analyst coverage, et cetera. So we already have a lot of interest from some of the, top ranked analysts in the United States, but it's very difficult for them to cover a company that's only listed on the TSX. So we feel good that we will have those relationships in place so that when we are listed on the US, we'll have that follow-on support.
Well, I'm a shareholder, and I view this, so I'm biased as anything. I view this as a unique opportunity where you are funded way above market values at significant amount of capital, which is different than so many other little biotechs in Canada that sort of pick up a few crumbs here and bootstrap along the way. And this is an opportunity for a retail Canadian investor or even smaller institution to buy it, what's essentially being funded like a private equity US-based big market cap, and you can buy it at a discount. It's not super liquid, but you can buy stock. So it's a very unique, well, as a shareholder, I'm frustrated by the share price. It does provide an interesting opportunity for investors, wearing my shareholder biased
Absolutely.
hat.
Absolutely. Yeah. Yeah. Once in a lifetime opportunity, we like to think.
All right. Excellent. We've been going on an hour now. We've gone through all the questions. That was excellent. Frank, I think you did a great job explaining everything and walking through that. So before we wrap this up, any final comments just to tie a bow on everything and sort of and concluding comments?
Great job, Martin. Thank you very much. Appreciate the questions from the audience. As you said and Liz said, you know, we genuinely believe that this is a once in a lifetime opportunity. We're doing everything we possibly can to move our drug along so that we get it as approved as fast as possible, but also as professionally as possible, because I and many others on the team have experience with trying to move things, you know, too quickly and getting caught out by that. We've delivered on everything that we said we would deliver on over the past several years, and we intend to keep that track record going. And, you know, we have something in the mechanism of action, the science, that is truly, truly unique. Comes along very, very infrequently in the medical and biotechnology worlds. It's very precious. And you know, we will get it into the market, and we will hopefully change the course of this disease and potentially other diseases. So, and we can't do it without the support of the market. So thank you very much.
Well, thank you very much for taking the time to give us all an update with this. It was greatly appreciated. Thank you very much. I look forward to watching the news as it comes out and to our further discussions.
Okay,
Have a great day.
thank you so
Thank you,
much.
Martin.
Okay, bye
Take
now.
care.